Thank you, Michael, and the rest of the conference organizers for inviting me to give this talk today. I'm excited to share with you some of my thesis work that I did in Tanya Henry's lab, specifically how we've used NMR to characterize and also troubleshoot designing dynamics in proteins. And I think it'll be a really nice follow-up talk. As you can see, we've also worked with calcium triggered switches but using computational protein design rather than a directed evolution approach. So I think it'll be a nice follow-up to the previous talks. And so, as I'm sure everyone in this room is well aware, proteins exist as conformational ensembles in solution, often adopting multiple functionally distinct states, where the bulk measured functional activity is often underpinned by the relative populations of these states. And in nature, redistributing these populations of states through perturbations like mutations or external stimuli or changes in the environment are often major modes of modulating and regulating protein activity. And so the goal of my work was to see whether or not those kinds of state-of-the-art design methods that Clara talked about earlier are accurate enough and generalizable enough to design this kind of behavior de novo. And so in specific, we aimed to see whether or not we could design proteins that interconvert between two user-defined structural states, and also see if we could modulate the conformational equilibrium through various perturbations. And so our approach for doing so was to first define the target states that we wish to interconvert between, and we can use those as input into a multi-state design algorithm, which aims to find sequences that populate both states at equilibrium. And these would be our dynamic designs that we can then experimentally characterize, which is what I'll be focusing on in today's talk. But we've also simulated these computationally using MD to try and get a better understanding of the molecular mechanism behind our observed bistability to tune populations rationally, but I won't be focusing on that as much today. And so when choosing our two conformational states to switch between, we had several considerations in mind. So the first thing was that we wanted them to be functionally distinct to not only enable experimental readout, but to also serve as a model system for being able to modulate function through changes to the conformational ensemble. And so for this proof of concept, we just picked something quite simple. So state 1 is calcium binding competent, whereas state 2 is calcium binding incompetent. And with this model, we should also be able to kind of test the converse idea of being able to use ligand binding to in turn modulate the conformational ensemble as well. And the second thing is that we wanted to explore a biologically relevant mode of motion. So here, to transition between states, this loop helix loop unit that I've highlighted undergoes a twisting and translation, but is relatively well packed against the rest of the protein that I'm showing in gray in both of the states. And I'll refer to this moving subsegment as the reshaped region during this talk. And I just want to note here and really emphasize that just because these kinds of localized motions, specifically localized rearrangements to secondary structural element orientations relative to one another, even though they're relatively subtle, they can often be functionally critical and highly evolutionarily conserved in large classes of biological regulators like kinases and GPCRs. And that's what motivated us to explore this particular mode of motion, which hasn't been explored in de novo design before. And so what do these states actually look like, and how do you generate them in practice? So for example, if you have a known binding competent conformation as state 1, what you could do is use a computational structural sampling algorithm to generate a library of de novo conformations for that reshaped region, and then through filtering for designability, come up with promising candidates for state 2, or you could do the reverse where you start with a known binding incompetent conformation for state 2 and basically use this structural library as scaffolds for ligand binder design. But because we're more concerned with designing motion rather than ligand binding, we started with a known binding competent state. So in specific, that's this natural calcium binding protein that doesn't undergo any appreciable changes to its conformation when it binds calcium. You'll see here I've highlighted the loop helix loop region in light blue, and it includes this calcium binding site, which is also the EF hand motif. And then with a computational sampling algorithm that was developed by Shinji Pan, who's a previous grad student in the lab, I generated this de novo conformation for the reshaped region, which you can see here highlighted in teal. And you'll notice that the calcium binding site has now been significantly reshaped to where it should no longer be favorable for binding calcium. But we can't just throw this artificially generated backbone straight into multi-state design without first verifying that it itself is designable, meaning that at least one sequence should be able to be identified that folds into this structure individually. So here you can just see the NMR structure of a single state 2 design that we solved with NMR with the help of Mark Kelly at the UCSF NMR facility, and it's in great agreement with our AlphaFold model. And so now that we've verified that both of these backbones for our 2 states are individually designable, we can use them as input in a much harder problem of doing multi-state design where we're trying to find sequences that can populate both states. And in today's talk, I won't really go over any of the multi-state design details, but basically we developed an approach where we generated dynamic designs that have very high sequence similarity, but diverse state populations with the intent of being able to more easily draw sequence dynamics inferences. So for example, we can more easily say this change to the sequence caused this change in the conformational ensemble for this particular mechanistic reason. And so with that approach, we ended up identifying this family of sequences that actually only differs at one residue position, which I've marked here by this red asterisk. It's in the central helix, which is actually quite distal from that calcium binding site, which is up here. And it seemed like depending on the amino acid identity at that position, AlphaFold predicted ensembles that were entirely in state 1 mixed or entirely in state 2. And just by grouping the amino acids based on their biochemical properties alone, we could see that it seemed like bulky hydrophobics at that position seemed to favor state 1 by pushing what we call loop 3 of the reshape region outward away from the central helix, whereas smaller hydrophilic hydrogen bond donors and acceptors seem to favor state 2, sometimes by forming this hydrogen bond with the backbone of loop 3, bringing it closer to the central helix into a conformation that was more consistent with state 2. And so we selected several of these designs for experimental characterization. So I'll just refer to them by the amino acid that's at that variable position. So if I say I-89, that's just an isoleucine at that position. But before kind of delving into the experimental results, I do just want to go over a couple of basic terminology and concepts related to chemical exchange. I know a lot of you are already familiar with this. But for our designed dynamic system, we're hopefully designing these sequences that are exchanging between 2 distinct conformational states. And so those would give rise, obviously, to 2 separate NMR spectra individually. And as a subset of these spins are kind of experiencing toggling between these different electronic environments, they're also have different resonance frequencies, and the difference in those resonance frequencies compared with the rate of exchange can affect what the NMR spectrum looks like. So if we're in slow exchange, you'd expect for a 2-state system for a spin to have 2 separate peaks resolved in the spectrum where the relative intensities are proportional to the state populations. Whereas in fast exchange, the chemical shift now becomes a population weighted average. And in intermediate exchange, where these quantities are comparable, you start to see the peaks broaden out as they are coalescing. And so with those concepts in mind, we did some very easy preliminary NMR experiments for screening to see if these designs were likely to be dynamic at all. And so again, they're only differing at that one residue position, but have different predicted state preferences. And so here you can see the N15 HSQC of 3 of our designs overlaid. And what we saw is for many sets of peaks, they seem to fall on a line. And so here I'm circling those sets in red, which could be consistent with systems in fast exchange, where you're seeing gradual changes to the chemical shift as the populations of states are varying between designs. And actually, when we later assigned the chemical shifts for I-89 and S-89, which seemed to lie on either end of the extreme, and then plotted those chemical shift differences on the structure, it seemed like there were high differences, obviously at and around the site of mutation, but also throughout the reshaped region, especially in that calcium binding site, which again is fairly distal to that mutated site. And so the next thing we did is we also collected a temperature series for the designs. And as you all are aware, there's usually a linear dependency between temperature and the chemical shift changes, but here we saw systematic deviations from linearity, some curvature, and also heterogeneous dependency of the intensity on temperature, which also suggested that there may be dynamics, and especially when we plotted the intensity of the peaks at 35 degrees Celsius compared to 5 degrees Celsius. We saw that these ratios were heightened in the reshaped region and in neighboring residues, which could be consistent again with the systems being in fast exchange and slowing more to an intermediate exchange regime as the temperature gets decreased where the peaks are sort of broadening out. And so with this promising preliminary data, we then decided to try and solve the structures of these designs with NMR. And so here I'm showing the structure for design S89, which again was predicted by AlphaFold to adopt state 2 with very high confidence. And you can see that the NMR ensemble in red agrees really well with our computational model for state 2. Whereas when we then tried to solve the structure for I89, which was predicted by AlphaFold to adopt state 1, but with notably lower confidence, NMR-TIS, which is the structure calculation software we were using, suggested that there were two distinct structures that could be consistent with our NMR data, one resembling state 1 and one resembling state 2. And so it seemed like by making mutations at this position, we were able to affect the conformational ensemble, where isoleucine at that position seems to have a much larger state 1 population as predicted. And then if you recall, state 1 should be calcium binding competent, so we should also be able to shift the conformational ensemble by adding calcium. And as Magnus mentioned, calcium is a really important secondary messenger, so we were really interested to see if we had coupled ligand binding to the conformational landscape. And indeed, when we saw the structure of iodine now bound to calcium, we're really just seeing one state consistent with state 1, which is preferentially stabilized by calcium. And so in summary, our structural data seemed like to suggest that we were successful in our engineering of these proteins that are exchanging between these user-defined structural states. And we can also modulate the populations both by mutation and by ligand binding. And so the next thing we wanted to experimentally verify was kind of the reverse of that concept, where mutations at this position, even though they're very distal from the calcium binding site, should be able to allosterically impact calcium binding affinity by shifting the conformational ensemble. And so we measured the KDs by NMR, and we found that making a serine to isoleucine mutation at that position does indeed have like a 10-fold impact on the measured KD. So it does seem like we're able to modulate this very simple function through changes in the conformational ensemble as well. And so the next thing we did, even though we didn't explicitly set out to design kinetics, was try to measure the time scale of our designed motion, mostly in response to a reviewer request. And so here I'm showing basically the phenomena that we're taking advantage of, where in chemical exchange, your subset of spins is experiencing stochastic changes to the resonance frequency, which causes enhanced transverse relaxation rates through dephasing, and you can quench this contribution of exchange to that R2 value through this class of NMR experiments called relaxation dispersion experiments. And by plotting the dependency of R2 on this dispersion strength, you can get really nice kinetic and sometimes thermodynamic parameters if you're in a favorable case. And so before trying to collect a full relaxation dispersion experiment, we just did a preliminary measurement of the R2 values for each residue and compared that to how much, basically a proxy for how much we would expect the local chemical environment of each residue to change just based on our structural models. It seemed like there was good correlation between elevated R2 rates and where we would expect bigger changes to the local chemical environment. And so this was promising, but then things got increasingly complicated. So as we've heard in some previous talks, there's different kinds of NMR experiments you can use to measure exchange times at different time scales. But you'll notice there's this like conspicuous void where in the supra tau C regime, it becomes difficult to quantitatively characterize kinetics at times. And of course, we fall into this void, mainly in this boundary region here at the low microsecond time scale. And so when you have this kind of mismatch between your sensitivity range of your experiment and where your exchange process is actually happening, some, as I found, unfortunate things can occur. So typically we would want to collect, this nice full relaxation dispersion curve where we're seeing this nice large change to R2 over our available range of refocusing powers. But since we're slightly too fast for the sensitivity range of our R1 row experiment, basically when we tried to collect data on this end of the curve with lower refocusing powers, there were a lot of technical complications to this, many of which involving some quantum mechanical explanations that people very kindly gave us, but I don't fully understand. But it seemed like essentially we were not able to align the magnetization properly throughout the course of the experiment, which resulted in these really large phasing artifacts, which cause, especially as you in the spectrum move away from the central carrier frequency. And so we were unable to reliably measure rates at this end of the curve. And as we go into the higher refocusing powers, obviously there's instrumentation limits. And so we don't want to burn out the probe either. So only a subsection of this curve was visible to us. And the next complicating thing was that when I was still optimistic that we were going to be able to reliably collect these curves, I collected them at several different temperatures in the hopes of eventually being able to calculate an energy barrier estimate. However, we saw that at lower temperatures, we actually had increased observed exchange rates, which was pretty counterintuitive. When we plotted the change in R2 for each position at different temperatures, we actually saw that there were some residues that had lower than baseline changes, and that was mostly in this loop and helix that were outside of the reshaped region, but spatially adjacent to it. Whereas within our reshaped region, we saw mostly elevated changes to R2 with temperature. And so this suggested to us that there were probably multiple exchange processes happening on different time scales. And that could potentially explain this sort of counterintuitive dependency between the exchange rate and temperature, where we might have a faster process that's becoming visible as we lower the temperature, as it slows down to fall more within the sensitivity range of our experiment. And so to try and test that hypothesis, we collaborated with Colin Smith, who is a lab alum as well, now a professor at Wesleyan. And here I'm just showing phi exchange, which is the product of the state populations and the chemical shift variance for each residue at each temperature, colored by, so basically he did a 2 times scale fast exchange model fit to our data. And so here the green corresponds to the faster exchange process, while the blue corresponds to the slower exchange process. And can be a little bit easier to understand if we zoom in on one residue. So here you can see in blue the contribution of the slower exchange process, and in green the contribution of the faster exchange process to our summed fit line in red. And it seemed like at lower temperatures, this faster process, which is localized to our reshaped region, becomes more visible, whereas at higher temperatures, the slower process seems to dominate and is more in that region that's outside of our reshaped region, but still spatially adjacent to it. And the time scale of exchange that we calculated for this faster process that's happening in our designed reshaped region is just an upper bound, since again, we were only able to reliably measure rates for a subsection of this curve. So it seems like we're in the low to mid microsecond regime, which is pretty typical for a motion of this kind. And so the last thing that I wanted to go over is just this anecdote about how we were also able to use NMR to diagnose undesirable dynamic behavior in our de novo designs. So this is work that I did in collaboration with another graduate student in the lab, Dominic Rosinger, who is leading this project. And so here you can see this is one of his dynamic designs, which is generated with a very similar method to what I described earlier. And when he first collected the N15 HSTC, he was really excited because there were all of these minor peaks that I've circled here in red, which suggested that there was a slow exchange process happening, which he hoped was arising from his designed motion. However, when he then went to collect these exchange experiments, he couldn't observe any exchange cross peaks, even at mixing times of up to 2 seconds, even at elevated temperatures. And so it seemed like this slow exchange process was happening very slowly, possibly even on the order of folding and unfolding, which would be really unexpected for, I guess just counterintuitive for a motion of this nature. And then when I assigned the chemical shifts for the major and minor peaks and plotted the magnitude of the difference, structurally, you can see they don't really fall within the reshaped region, which are these two helices on the left, but they're much higher in this beta strand. And so it turns out when we then collected some nosy data, it seemed like those minor peaks were not arising from his design motion, but actually from two different conformations of this beta strand. So here you can see in gray, one of the conformational states for this beta strand was as we designed, whereas the other in blue is shifted down by one residue. So all of the amino acids are basically facing the opposite side of the beta sheet. And that was likely due to this like unintentional symmetry in the beta strand sequence. And so with this information, he was then able to make a point mutation to destabilize that undesirable beta strand conformation. And you can see here the original design N15HTC in black and this point mutant in blue. And now we've collapsed just basically to one state. And so in conclusion, there's a lot of different ways that we can use NMR to help us characterize and troubleshoot de novo design dynamics in proteins, including these quick screening for dynamic designs. In my case, I was able to use the nosy data to resolve multiple structural states, which I know isn't typically common, but in some favorable cases you can. It's also useful for characterizing binding affinity, especially in our case where we had there's actually multiple calcium binding sites of comparable affinity, but we only redesigned one. It can be useful for kinetics, although obviously there are complications that can happen if you're not on the right time scale or there's multiple exchange processes going on, which is quite common for natural proteins. And also we can use it to help troubleshoot design dynamic behavior as well, but obviously that requires a lot of human time and labor. And obviously this is all under certain conditions. So with that, I'd just like to thank everyone in the Pertemin lab, especially my collaborators, Don, Denise, Shinji, Colin, and a huge thank you to Mark Kelly, who I think some of you might also know, but he runs the NMR facility at UCSF and taught me all the biomolecular NMR that I know. And this is my cat, who also spends a lot of time looking at M15 and for NMR spectra with me. So thank you all for listening.